Hello, in this video we will discuss about the iron physiology. So we will just understand the overview of the iron physiology, and we will discuss about the function of the iron. So basically the iron is very important in our body, and daily iron requirement is 10 to 20 milligram. So the iron is basically when we will take that is in the form of ferrous, and that will need to convert ferric its plus 2, so that is the cation. So here is the stomach. So let's begin to understand the iron function. So here when we will take the food, for example the vegetable or other protein source which that may contain iron with the quantity on the basis of their quality of food. So when we will eat the that ferric basically is go into the duodenum first, and the target for the absorption of the iron is the duodenum and the small intestine, duodenum, ileum and jejunum. So here I am taking the duodenum. So here is the lumen of the duodenum, which that is I am describing, and contain enterocytes. This enterocyte is also known as the brush border cell. This brush border cell contain a brush border enzyme. For example, here is the border of the brush contain a vitamin C ferroreductase enzyme. This vitamin C ferroreductase enzyme will convert the ferrous into the ferric, and this conversion is able to move into the enterocyte. So when move into the enterocyte, it will store either into the ferritin protein, which that is in the form of globule, and on by the conversion of again ferrous, while on the other hand it will be transport into the blood circulatory system. So here is when move inside of the enterocyte by the symport, it is the divalent metal transferase 1 enzyme, channel is the DMT1. So when move with the symport transport, it means the hydrogen ion will be also moved. It means the proton is used for it. So that is known as a symport. And use this ferric is go into the ferritin globule or move into the interstitial space by the ferroprotein protein containing channel, which that is after moving into the interstitial space, it will again convert into the ferrous. Why? Because that is the shuttle mechanism, and it means the ferrous convert into the again ferric convert into the again ferrous, and this conversion of the ferrous to ferric will lead to move into the circulatory system, where the important thing is that the apoferritin protein, which that is used for the movement of the iron with the binding. So it means the two ferrous will be bind with the apoferritin protein, apoferritin. So after the binding, it will be function two types. Basically, it can go into the bone marrow for erythropoiesis. So it means that 75% go into the store into the bone marrow and the bone marrow formation, and it is also play important role for erythrocyte formation. Basically is the hemoglobin formation in the erythrocyte. So the transferritin receptor are present in the erythrocyte, which that is engulf the iron. So the erythrocyte are red blood cell. Basically, when take the iron, it means the hemoglobin central unit will be produced. So that is erythrocyte will become a red blood cell mature after the absorption of the iron in the bone marrow. So it is the developmental part is the origin of the development of erythrocyte into the red blood cell are present in the bone marrow, red bone marrow. So the hemoglobin carrying oxygen for carrying the oxygen and carbon dioxide for respiration is important, the red blood cell capacity. So here is the liver. The second one is the movement of the apotransferrin into the liver, and it means it give away the ferrous to the liver, where the transferrin receptor are present, which that is transferrin receptor is allowed to move 10 to 20% ferrous, and that is the ferritin protein either stored into the liver, or basically it's again move into the bone marrow again. That is the major reason. But important thing is that the liver is also play important function for storing of the iron for further requirement. If the less requirement when we will not eat during the fast state, so the iron will be released from the liver. So here I am drawing the liver diagram, hepatocyte, which that is the extracellular fluid of the here intracellular fluid of the hepatocyte, and that is the left and right is the extracellular fluid. I am drawing just a cross section. So here is the transferrin receptor are present, which that is the diamond. When bind with the two transferrin protein bind and to engulf, it is known as the endocytosis. So it vesicle will be formed. This vesicle will be accept the proton to decrease the pH, and after the decreasing of the pH, it means after the decreasing of the pH, it is play function 2 type, expression of DMT and the detachment of the transferrin receptor. After the detachment, basically when detachment, so the transferrin will be released, while the ferrous will be again converted into the ferric, and this conversion basically again ferric to increase the ferric, increase the ferric, which that is stored into the liver or transport basically to another tissue for the requirement on the basis of the requirement. So important thing is that here is the ferritin will be accept that. And on the other hand, the important thing is that I am discussing in this way here is the transferrin transport receptor basically will bind this again for further accepting of the transferrin protein for the accepting of the iron. So the extracellular fluid basically is again absorb transfer the transferrin into the blood circulatory system. So here is the uh, hepcidin. Hepcidin is important thing is that it is play a functional role. Hepcidin is the master iron regulator, which that is play a homeostasis formation. When hepcidin will go into the blood vessel, blood circulatory system, so it means the hepcidin will be inhibit the uh, uh, ferroportin channel. So that is inhibiting is the ferroportin channel for so that is inhibit the absorption of more iron. Why? Because the excess of the iron are present in the liver, so we know we should not need to absorb the more iron. So on the other hand, the hepcidin is also inhibit the releasing release of the iron into the splenic macrophage. Basically here I am discussing about the macrophage is engulfed the in the spleen to old red blood cell to break down to produce more ferric. So this mechanism will be inhibited due to the excess amount of the iron in the red blood in the blood circulatory system. So the ferroportin will be also inhibited by this mechanism. So that is why the hepcidin is the inhibitor or block of the ferroportin for the transport of the iron into the blood circulatory system, and the release into circulation it will be inhibited. Why? Because the excess of the iron are present into the circulatory system, it means we will not need any more. So when decrease the blood circulatory system, iron will decrease in the blood circulatory system, will lead to stimulate this, and hepcidin will be decreased. So this hepcidin is basically another is the HFE which that is create, and this is the hematochromatosis gene which that is stimulate, and this hematochromatosis gene basically is the uh, mm, protein HFE which that is lead to mutation, and this mutation is basically lead to increase the hepcidin. So this hepcidin will be increased due to the protein HFE which that is produced in the liver hematochromatosis gene which that is produced, and the plasma ferrous will be increased which that is plasma ferrous increase to lead in increase the hepcidin, and this hepcidin increasing of this hepcidin will stop the breakdown of the red blood cell and stop the increasing of the iron. So on the other hand, the plasma iron concentration will be decreased, and on the other hand, the lipopolysaccharide which that is also stimulate the hepcidin to increase to decrease the iron which that is the bacteria produce. So here is the basically also inhibit the ferroportin one receptor basically basically is the IREG1 receptor. Basically it is inhibited for the further absorption of the iron. So it means that is the hepcidin is the regulator of the iron absorption and the circulation into the blood circulatory system for the normal function. So this is the hepcidin is important homeostatic protein which that is play important function as a homeostasis. And more here is the vegetables and hemoglobin and myoglobin in the dietary myoglobin and hemoglobin which that is come from the meat, and the vegetable which produce the heme and globin after the digestion in the duodenum. This heme will be entered by the HCP1 possible channel, and after move into the heme inside, it means the bilirubin formation and the iron ferrous formation, and this ferrous is basically the 3 positive charge. So that's move inside, and the bilirubin basically and iron ferrous will be again absorbed if the requirement. If not requirement, so hepcidin is inhibited by this channel. It means the ferroportin will be inhibited by the hepcidin. So that is the major mechanism. While the DMT1 is also used for copper, zinc, magnesium, and other metal ion for transport, which that is also our requirement in our body for the normal function. So that is why, for example, the zinc is also play important role for the production of the androgen as well as the sexual hormone. On the other hand, the zinc is on the other hand, the magnesium is also used for the translation of the protein by the binding of the ribosome. So here is the female basically is a very controversial problem for the menstrual cycle, which that is increase the loss of hemoglobin and protein, and the increase of the blood ferrous. So it means decrease of the blood ferrous, and the 50% iron more required as compared to the male, and the hemoglobin decrease, it means the anemia situation or blood volume loss, and the ferric is basically produce a free radical to cause the cancer. So that was the and mutation also. So that was the overview of the iron physiology. I hope you make sense. Thanks for watching. Please share this video and make sure to subscribe, like.